Okay, so today we are actually we're in the week of October 19th through the 22nd. We've already finished muscular system. So we're going to cover neural tissue starting this week. It's a lot to cover, so it'll probably take us 2 weeks. We have a mastering A&P quiz this week on muscle physiology. We have, I think you've already taken quiz 3, but then we have exam 3 over chapters 9 through 11 at the end of the week. So I want to make sure you see that. Yes. I'll get it posted later today. Thank you for letting me know. All right. So since we're starting a new unit, show you the exam review. So essay questions, explain the steps in the generation of an action potential, explain the steps and the function of a cholinergic synapse. So you probably don't know what those are today, but we'll get to them in this chapter. Short answer, fill in the blank. 2 functions of the nervous system. Difference in action potentials between neurons and muscle tissue. List 1 reason active neurons need ATP. Who's faster at responding to stimuli, the brain or the spinal cord. List 1 state of gated channels. List 1 of these steps that's involved in the transfer of information. Medical problems that would result from damage to specific parts of the nervous system. And let's see, for matching the different nervous system diseases, different types of neurons, and stages and parts of an action potential. Multiple choice. Why are sodium potassium channels open and closed during action potentials? Cholinergic synapses. Divisions of the nervous system and their functions. Locations and functions of glial cells. Types of neurons. How a reflex is classified. Types of reflexes. Sequence of a reflex arc. Parts of the brain and their functions. Primary brain vesicles and what they develop into, neural folds, meninges, medical conditions of the brain and spinal cord, parts of the neuron, parts of the synapse, functions of cerebral spinal fluid. Terms you need to know: refractory period, threshold stimulus, resting membrane potential, all or none principle, myelin, saltatory transmission, action potential, depolarization, repolarization, hyperpolarization, gray matter, white matter, dura mater, pia mater. Okay. So cerebral spinal fluid. All right. So it just gives you an overview of what we're going to be talking about in this unit. Okay. There is a document. So if you go to the modules for week 11, it says nervous system functions. So if you click that, it'll take you to this document. So you will have some questions asking you about the functions of the nervous system. So you need to be able to explain if the nervous system directs immediate responses to stimuli, coordinates or moderates activities of other organ systems. It provides and interprets sensory information about external conditions. Okay, so you need to be able to explain that. Okay, I want you to know where that is. All right. So let's go ahead and start the chapter. Okay, so now we're moving into the nervous system. Okay, you got different organs that are involved. You have your brain, your spinal cord. You also have receptors for sensory organs, okay, like your eyes and your ears. The nerves connect to other systems. Now, if we're thinking on the tissue level, okay, you have, remember, tissue is composed of cells, right? So the cells involved in nervous tissue, you have the neurons. And neurons are actually considered cells, and they're used for communication from one cell to the next. We also have neuroglia, or glial cells. Okay, and these help neurons survive and function. And they help preserve the structure in your nervous tissue. Okay, so you have the neurons and the neuroglia that support them. Okay, we're going to separate. We're going to separate the nervous system into two parts. You have the central nervous system and the peripheral nervous system. So what's the central nervous system? Well, it's your brain, your spinal cord. It's made up of nervous tissue, connective tissue, and blood vessels. What does it do? Well, it coordinates and processes information or sensory data from inside your body as well as outside your body. Okay, that should make sense. Sends motor commands that control activities in other organs like your skeletal muscle. So the brain specifically is involved in what we call higher functions. Things like emotions, learning, memory, and intelligence. Okay. Okay, so the peripheral nervous system is basically all your nervous tissue that is not a part of your central nervous system. And it's going to provide or send information to the central nervous system. It's also going to send these motor commands to the tissues. We call them peripheral tissues because they're distributed throughout the body. So nerves, particularly peripheral nerves, are just bundles of axons. Okay, and we'll talk about the structure of a neuron a little later. But they're bundles of axons. Okay, and also including connective tissue and blood vessels. And nerves can carry both sensory information as well as motor commands. So they can receive sensory information, and they can send signals, motor commands. Or they can send sensory information to the brain or part of the nervous system, or send motor commands to peripheral organs. The cranial nerves, as the name suggests, connect to the brain. Spinal nerves are going to be attached to the spinal cord. Okay? So the nerves can be cranial or spinal. Now, that's sort of describing the nervous system based on structure. Now let's talk about dividing up the peripheral nervous system based on function. So the peripheral nervous system has an afferent division, which is going to carry sensory information. And that information comes from receptors in your various tissues, as well as organs. And that information is going to be sent to the central nervous system, to the brain and spinal cord. Now you also have an efferent division, and that's responsible for carrying motor commands, telling your body to do something. So here information is going from the central nervous system to your muscles, could be the adipose tissue, could be the glands. Okay. So afferent is sending signals to the central nervous system. Efferent is sending signals from the central nervous system. Okay. Now, we also have structures called receptors, and their role is to detect some changes in the environment around it. Could be to respond to some stimulus. These receptors could be neurons. They could also be some specialized cell in your body. Receptors can actually be an individual cell or on an individual cell, or they can be a whole organ, like your ears or your eyes, okay, our sensory organs. You also have effectors, and these effectors are going to target organs, and those organs are going to respond to some motor commands. So in a sense, the receptor is where the signal is coming from originally. The effector is essentially where the signal goes after it's been to the central nervous system. Okay. So basically information goes from the receptor to the central nervous system. Another signal goes from the central nervous system to the effector. Okay. Now for the efferent division, you can divide that into the somatic nervous system and autonomic nervous system. So your somatic nervous system is basically things not necessarily all conscious, because there are some involuntary functions like reflexes that are in the somatic nervous system, but a lot of it is voluntary, hence skeletal muscle contractions. Okay. The autonomic nervous system handles things that are more subconscious, things that you're not really thinking about. So things like contractions of your cardiac muscle and smooth muscle, you don't have to think to make your heart beat, or your blood vessels, the arteries move blood through your body. You don't have to think to make your intestines move food through your body. You also have glandular secretions that are autonomic. Okay, things you don't have to think about. You don't have to think to make hormones. Okay, now within the autonomic nervous system, when you think autonomic, think more automatic. Could be, there's a sympathetic division and a parasympathetic nervous system. Typically, the sympathetic nervous system does things that stimulate. Typically, the parasympathetic nervous system is going to do things that relax. Okay. All right. We also have an area, a part of the nervous system called enteric. You have millions and millions of neurons in the walls of your digestive tract. So it's many or more than you would even have in your spinal cord. You also have neurotransmitters like the ones you have in your brain. So it's involved with visceral reflexes, and they don't need information or instruction from the central nervous system, but they can be influenced by the autonomic nervous system. Okay. And so here's a visual representation of that. Again, you have your central nervous system, your brain, your spinal cord. Then you have your peripheral nervous system, okay, nerves and nervous tissue. outside your central nervous system and your enteric nervous system. You have an afferent division and efferent division. Sensory information is within the afferent division. Motor commands are within the efferent division. So for the afferent division, you've got essentially somatic sensory receptors to deal with things like your skeletal muscle, joints, and your skin surface. Visceral, which has to do with the organs inside your body. And special, really like your special senses like smell, taste, vision, balance, and hearing. Okay, but in all of these cases, you're sending information to your central nervous system. The efferent division, again, we're sending motor commands to the tissues. With the somatic nervous system, deals with skeletal muscle primarily. Autonomic can be separated into sympathetic and parasympathetic. And it acts on smooth muscle, cardiac muscle, glands, and adipose tissue. Okay, so this image is also in the e-text. So if you haven't looked at the e-text in a while, I would highly recommend you be looking at it. More successful folks in this class read the e-text a lot. So I would highly encourage you to make that time. I mean, it's great to just skim the full notes, but e-text is really helpful. All right, so let's pause, and I want you to talk with your partner about the divisions of the central, or divisions of the nervous system. All right, and you can do this at home as well. Okay, so let's talk about neurons. So neurons are your basic functional unit inside your central, or inside your nervous system in general. Okay. What do? They receive signals and they send signals. And they do different things involving control of other parts of the body, processing information, and communicating. So some of the parts of a neuron, your cell body, also known as a soma. Okay, it has a large nucleus and nucleolus. Remember, the nucleolus has to do with creating the ribosomes. The cytoplasm is called the perikaryon. We know mitochondria are involved in making energy. We know that roughy R and ribosomes are involved in making proteins, but here they're in the context of what you need for a nervous tissue cell. All right, so the cytoskeleton, remember that structural element in the cytoplasm, in this case we call it a perikaryon. It's made up of some structures like neurofilaments and neurotubules. And they are similar to what we see generally in cells with the intermediate filaments and the microtubules. The neurofibrils are basically bundles of neurofilaments. So just think levels of organization here. So if you take a bunch of neurofilaments, you can get a neurofibril. And these support the dendrites and the axons, which we'll talk about in a moment. You also have Nissl bodies. These are areas where you'll see roughy R and ribosomes in the cytoplasm. It's very dense, it's a highly packed area of ribosomes and roughy R. And that's what makes your nervous tissue look gray. When we talk about the gray matter, we're talking about the Nissl bodies. So the dendrites These are short branch extensions from the cell body. Now, in the dendrites, we have what we call dendritic spines. And these are just little processes on those dendrites. And they're able to get information from other neurons. But they actually are a large component of the neuron surface area. Okay, these spines add surface area, which is important because then it allows more connection between neurons. Now, the axon is a long process that has cytoplasm in it. You usually just have one. You can have many dendrites, but usually just one axon. And its role is to propagate or conduct an electrical signal. And happens in the form of action potentials. Okay? So we talked briefly about action potentials and we talked about stimulating muscle tissue. All right, so now we're going to take a step back and see how that action potential works in neural tissue. Okay, in the axon, we call the cytoplasm axoplasm. And in that axoplasm, you'll find organelles, you'll find enzymes, you'll find neurotubules and neurofibrils. Okay. So here's a visual representation of a neuron. Okay, so you see the cell body or the soma. Inside you got the nucleus and the cytoplasm. Remember we call it perikaryon. Here are the dendrites. You can see all these dendritic spines along the ends. Here's your axon. And the endings we call telodendria. Okay. Now, important distinction here. So axons are going to send information, dendrites receive information. Okay, so important point. You make sure you know that. Okay. All right. All right, so in the axon, just like the cytoplasm is the axoplasm, the plasma membrane is the axolemma, excuse me. So remember, in the muscle tissue, the plasma membrane was called the sarcolemma, okay? And the axon is called axolemma. And it provides a level of covering over your axoplasm. The base of the axon is known as the initial segment of the axon. And the axon hillock is your region that's very thick and attaches the initial segment to the cell body. Okay. All right, so here's kind of a visual of that. Here's your axon hillock and your initial segment. Okay. All that plasma membrane in the axon is axolemma. The endings of the telodendria are called axon terminals. The space or connection between these two neurons is called a synapse. So the one that is sending information is the presynaptic cell. The one that's receiving information is the postsynaptic cell. Okay. Here's your nucleus, your nucleolus, your Nissl bodies, like your Ruffy-R and ribosomes. Okay. Here's some neurofilaments. There's a Golgi apparatus. These are all your dendritic spines or branches. Okay. And overall, This is the direction that the signal is traveling or propagating. It's the direction of the action potential. Okay, axons can have branches, and we call those branches collaterals. Again, those extensions of the axon on the distal ends, okay, toward the end are telodendria, and then the tips of the telodendria are axon terminals or synaptic terminals. So if you're moving materials between the cell body and the axon terminal, we call that axonal or axoplasmic transport. So the materials are going to then move along the neural tubules within that axon. And it takes energy to do this, typically. So the mitochondria are going to be involved, as well as the proteins kinesin and dynein. Okay. So if we want to classify neurons structurally, we can say they're anaxonic, which means they're going to be small. All the cell processes are going to look alike. You see these in the brain and some of your special sense organs. If they're bipolar, it means you have two poles, right? So you have one axon and one dendrite. And in your special sense organs like sight, smell, and hearing, that's where you'll see that. Now, if there's only one pole, it's unipolar. Okay, so the axon and your dendrites are actually fused together, and you have a cell body on one side. And your sensory organs and your peripheral nervous system are mostly unipolar. Okay, so if you have one long axon but multiple dendrites, it's said to be multipolar neuron. These are common throughout the central nervous system. All those motor neurons that control skeletal muscles are multipolar. All right, so what does that look like? Here's your anaxonic neuron, more than two processes, could all be dendrites. All right, sometimes the axons are not obvious. Here's your bipolar neuron, two poles. Okay, one axon, one dendrite. Unipolar, and you might say, well, isn't that the same thing as bipolar? Well, it's not, because the axon and the dendrite are fused together right here, and they both come together to go to the cell body. Okay, so you got to make sure you look for that. Also, on the bipolar, the extensions go from opposite sides of the cell body. Okay, but if they're coming into the same point on the cell body, it's unipolar. Okay. Now here you got multiple dendrites in one axon, so it's multipolar neuron. Okay, and we've gone through this already. So let's pause. I want you to talk about the structural classification of neurons as well as the structure of a neuron. You can do that with your partner. Okay, so that was the structural classification of neurons. Let's talk about functional classification of neurons. So we have sensory neurons, motor neurons, and interneurons based on function. Sensory neurons are afferent, okay, because they're sensory. They're typically unipolar. And so their processes, known as afferent fibers, are going to go from the sensory receptors somewhere in your body to the central nervous system. Now, if their sensory neurons are somatic, they're monitoring the external environment, and if they're visceral, sensory neurons are monitoring the internal environment. So that tells you, so if you're trying to learn the words, what does somatic mean? External. What does visceral mean? Internal. So we tend to see somatic associated with skeletal muscle. And although that is technically not external to your skin, but it's more external than your digestive organs or your heart or something like that. Okay. So think about it that way. Now, there's different kinds of sensory receptors. You have enteroreceptors, exteroreceptors, and proprioreceptors. So if you're monitoring internal systems, things going on inside your body, you have enteroreceptors for that. So things like your urinary system, digestive system, or internal senses. So this would include things inside your skin like stretch, deep pressure, or pain. But we're typically talking about not really the skin. We're talking about stretch or pressure or pain inside your body. Okay. Whereas the exteroreceptors monitor things going on outside your body, like how hot or how cold it is, the temperature. Also things that you see, smell, and hear. Okay. So you use exteroreceptors for that. Now the proprioreceptors have to do with your skeletal muscles and monitoring in your joints and monitoring the movement of those muscles and joints as well as their overall position in the body. Okay. So the enteroreceptors, exteroreceptors, proprioreceptors. All right. So we just talked about sensory receptors. Now let's talk about motor neurons or sensory neurons. Let's talk about motor neurons. Motor neurons are efferent, right? Because they're basically sending information from the central nervous system to some effector in the body. And they use axons or efferent fibers to do that. If the motor neurons are somatic, they innervate skeletal muscle, right? Part of the somatic nervous system. If they are visceral motor neurons in the autonomic nervous system, basically they're innervating other effectors. All right. So things like adipose tissue, cardiac muscle, glands, smooth muscle, okay? These are all visceral motor neurons, okay? And they are causing a function that is part of the autonomic nervous system, okay? These are things that are gonna happen without you having to decide to do them. All right, so the motor neurons. So these are signals from your central nervous system, and they're going toward the visceral effectors. And they're crossing what we call autonomic ganglia or nervous system cells. And they're going to divide axons into what we call preganglionic fibers and postganglionic fibers. All right, so with your inner neurons, most are going to be in the brain and the spinal cord. But you do have some in autonomic ganglia. And where you're going to see these is in between sensory neurons and motor neurons. So that's the big difference. An interneuron, if you have one, is in between a sensory neuron and a motor neuron. Okay, that's like the big picture thing, all right? And they can help distribute sensory information. They can help coordinate motor activity, help you with learning, planning, and memory. Okay, but the big thing is they're in between a sensory neuron and a motor neuron. All right. So let's pause and describe sensory neurons and motor neurons, two partners. You can do this at home as well. Okay, with the neuroglia, remember these are our supporting cells. They support and protect the neurons. Over half of your volume in the nervous system is made up of neuroglia. And there's many different types of neuroglia. All right, and they exist in both the central nervous system and the peripheral nervous system. All right, so in the central nervous system, you've got astrocytes, you have epinemal cells, you have oligodendrocytes, and microglia. All right, so here's what the astrocytes look like. So they look kind of like stars, okay? If you're kind of creative, and they're involved in maintaining blood-brain barrier, okay, regulating ion, nutrient, gas concentrations, forming scar tissue after injury, absorbing and recycling neurotransmitters. The epinemal cells they're going to line your ventricles in your brain. We'll learn about what those ventricles are, as well as your central canal and your spinal cord. And they help with making, circulating, monitoring your cerebral spinal fluid. Your oligodendrocytes, they provide myelin for your axons in the central nervous system. They'll be wrapping around the axons. Your microglia basically get rid of all your cellular waste or debris, and also pathogens, and they work through phagocytosis. Okay, that's it. So you can see the microglia. And so the astrocytes have like these little knobs on them, whereas the microglia have kind of like little, they're like pointy on the ends. Okay, so make sure you can see the difference. So a little more detail here, the astrocytes. So their cell bodies are large, lots of processes, help maintain your blood-brain barrier. Big thing that astrocytes do. Okay, they can help repair tissue. All right, help neurons when they're developing. All right. So here's some myelinated axons. We'll talk more about those myelin in a little bit. But these cells that produce that are your oligodendrocytes. And here's an astrocyte. Has lots of these little knob projections on it. Okay. Star-shaped cell, anchored to capillaries, maintains blood-brain barrier. Okay. This is all what we call the white matter of the central nervous system. All right, so our epinemal cells, these are going to form an epithelium. It aligns your central canal of your spinal cord, as well as the ventricles in your brain. Big thing is that they are involved with cerebral spinal fluid. okay? Make it, monitor it, circulate it. All right, so here's another picture. Here are your epinemal cells, simple cuboidal epithelium. Okay, one layer of cuboidal cells. They line fluid-filled passageways. Okay, another type of cell here, microglial. These are phagocytes. And they go around and they basically eat up debris and pathogens, get rid of them. And the epinemal cells and the microglia, you see those in the gray matter, okay? Okay, your oligodendrocytes, you form the myelin sheath, insulate your myelinated axons, and that is important because they make action potentials go faster. Okay, so the signal gets sent much faster. Okay, so it gives nerves their wide appearance. Okay. All right, so relative to the oligodendrocytes, again, if the segments of the axon have myelin on them, we call those internodes. And then we also have nodes, or nodes of Ranvier, that are in between the internodes. Okay, you can also have branching of axons there. And we'll see later when we learn about action potentials that the signal travels from node to node. Okay, so it's skipping over the internodes. All right, you have white matter and gray matter. So white matter, so these are regions of your central nervous system with lots of myelinated axons. That's why they're white. Gray matter has the unmyelinated axons, as well as cell bodies of neurons and dendrites. Okay, so the white matter is white because of the myelin. Okay, so looking at this again, here's your oligodendrocyte, here's your myelin, so it's in your white matter. Okay, the microglia is your smallest, least numerous. Lots of branches, they're very fine. Again, phagocytes clean up waste, debris, and pathogens. Okay, you can see that here in the gray matter. Okay, so that was the central nervous system. So let's take a minute, let's pause and talk about the neuroglia in the central nervous system. So let's talk with your partner about that. You can do this at home as well. All right, let's talk a little more about neuroglia this time, in the peripheral nervous system. We've been talking about this neuroglia in the central nervous system. Now it's time to talk about the neuroglia in the peripheral nervous system. Okay, so what do? Well, they're going to insulate the cell bodies in the neurons, as well as the axons. We only have two types in the peripheral nervous system. We have satellite cells and Schwann cells. All right. So our satellite cells, so they're going to surround the cell body of the neuron at a ganglia, or collection of neurons, or collection, excuse me, of neural tissue. They're going to regulate levels of neurotransmitter, nutrients, oxygen, and carbon dioxide around those neurons. Now the Schwann cells are going to surround all of the cells in the peripheral nervous system, and they help myelinate the peripheral axons. They're also going to help participating in repair if you have an injury in your peripheral nervous system. Satellite cells and Schwann cells. All right, so the satellite cells, they regulate the fluid around the neuron. Okay, surround clusters of cell bodies called a ganglia. Okay, the Schwann cells form myelin sheath around the axon. All right, so just like the plasma membrane around the axon, we called it axolemma. The outer surface of a Schwann cell, we call that a neurolemma. And a Schwann cell will only provide a myelin sheath for one axon. But many Schwann cells actually are going to provide a sheath for an entire axon. Okay, so here's an example of the Schwann cells providing myelin for this peripheral nervous system axon. Okay, here's the nucleus of the Schwann cell. Here's some myelin covering the internode. It's wrapping layers around it. Here's the axon. Okay. Outer covering is the neurolemma. So this whole thing forms the myelin sheath. Okay. All right. Here's your Schwann cell. It's a big nucleus. Neurolemma is surrounding it, and it's wrapping around all of these axons. Okay. So it's forming essentially a bundle of nerve fibers. All right, so it's your nucleus, your Schwann cell. All right, but here are these individual axons. Okay, here's the neurolemma surrounding all of them. So how does that myelin sheath form? So your Schwann cell is going to surround a portion of an axon within that groove in the cytoplasm. Then that Schwann cell is going to rotate around the axon. And you wrap or keep wrapping around, forming multiple layers. You get a tight packed membrane. Okay, so get an injury to your neurons. Axon could degenerate. Schwann cells will wrap around a new axon by forming a pathway for new growth. Okay. It's difficult to regenerate your nerves in the central nervous system, and that's because of the astrocytes. And they produce scar tissue, and they release chemicals that actually block regrowth of the neuron. So that's part of why getting damage to your nervous system is so bad, because it's hard to regrow the neurons. All right. So here at this injury, you get fragmentation of the axon. Schwann cells form some sort of cord, and they grow into the cut and unite the stumps or the ends. Macrophages come in and engulf the myelin and the degenerated parts of the axon. All right, axon's going to start sending buds into this network of Schwann cells, and it starts growing along this cord of Schwann cells. Axon continues to grow to the distal stump. It's enclosed by a Schwann cell. Okay. All right, so let's talk about our neuroglia in the peripheral nervous system. So I'll give you a minute to do that. All right, so all plasma membranes are going to make electrical signals through movements of ions. The neurons really are basically, they really rely on this membrane potential. Okay, it's very important. And so we have different types of potentials that we talk about with neurons. We have a resting membrane potential, and it just means the potential of a cell when it's at rest. You also can have a graded potential. It's temporary and specific or localized change in the resting potential. And that happens when you've been presented with some stimulus. An action potential generally, basically it's an electrical impulse, happens because of the graded potential, because of that change. And it's going to move or propagate along the axon on the surface. And it's going to move toward the area called the synapse, okay, the ending of the axon terminal. So that resting membrane potential. So there's three important concepts here. So there's a big difference in composition between the extracellular fluid and the intracellular fluid, okay? So in your extracellular fluids, you have high concentrations of sodium, high concentrations of chloride. In your cytosol, you have high concentrations of potassium and other negatively charged proteins. The cells there their membranes are said to be selectively permeable. And the membrane permeability varies, okay, depending on the ion, okay? Okay. Okay, so there are processes that happen across the cell membrane that do not require energy. They're passive. So the current is basically charge moving and is trying to get rid of a difference in potential across the membrane. So that's current in the context of a neuron. It's flow of charge. Resistance has to do with the membrane restricting the movement of those ions. So if you have a lot of resistance, that means the flow of charge is small, or the current is small. If resistance is low, then the flow of charge is high, or the current is high. Okay. So here you've got a difference in charge. Okay, more potassium on the inside of the cell, more sodium and chloride on the outside. Okay, so you end up getting a concentration gradient or chemical gradient. Okay. And to maintain that, use the sodium potassium pump, because naturally through diffusion, you're going to be moving charges across the membrane. And you have sodium potassium pump to restore or maintain the membrane potential. All right, so we can actually use machines to be able to measure the potential difference. Resting membrane potential is negative 70 millivolts. Okay, so more on these passive processes. You have a chemical gradient, which is basically a concentration gradient dealing with ions. Electrical gradient has to do with charges that are separated, the difference in charge. So the cytosol is considered negative relative to the extracellular fluid. Okay. More negative charge on the inside of the cell relative to outside. Now, if you combine or sum all of the chemical and electrical forces, they're acting on some ion across the membrane, we call that electrochemical gradient. And it serves as a form of potential energy in the sense that that difference in charge could have a result. So it's potential energy. Now, equilibrium potential means there's no net movement of the ion across the membrane. It's at equilibrium. So for potassium, that's at negative 90 millivolts. And for sodium, it's positive 66 millivolts. Okay. But the plasma membrane is actually more permeable to potassium ions than sodium ions. Okay. So that's why the resting membrane potential is closer to the equilibrium potential for potassium. So the resting membrane potential resting membrane permeability of the sodium ions is low. So your sodium ions don't have a big effect on the resting membrane potential. All right. So at resting membrane potential, your chemical and electrical gradients are basically opposed. As you have more potassium, higher concentration of potassium inside of the cells, that gradient's going to essentially force potassium ions out of the cell. Okay, high concentration and low concentration. And if you were to continue to allow that to happen until you got to equilibrium potential, where there's no net movement, you'd be at negative 90 millivolts. For sodium, higher concentration outside the cell in the extracellular fluid, net movement, or the direction of movement would, for the electrochemical gradient, would force sodium inside the cell. But it's more difficult to move the sodium inside the cell. So you have a huge swing in order to get to equilibrium potential. Okay, goes all the way to positive 66 millivolts. Okay. All right. Now, we talked about passive processes. Here's an active process. That's the sodium-potassium exchange pump. It's active because it's going to be powered by ATP. All right, so it's going to basically move 3 sodium ions out of the cell for every 2 potassium ions brought in. And that balances the passive forces of diffusion because it's essentially going the opposite direction. Because remember, potassium wants to go out of the cell. Sodium potassium pumps bringing it in. Sodium wants to go into the cell. Sodium potassium pumps moving it out. It helps keep your resting membrane potential stable. And that's happens when that ratio of sodium entry to potassium loss, okay, through the passive channels is 3:2. So it has to balance out. If that is what's happening passively, sodium potassium pump can balance it out. Okay. So I'm going to pause there, allow you to do your exit tickets, and then we'll pick up tomorrow, or Thursday, talking more about the resting membrane potential. Okay. All right, so we'll start here next time.